Welcome everybody to Gaining Insight on the INSIGHT Phase 3 clinical trial. During this seminar, all participants will be in a listen-only mode. There will be a question and answer session at the end of the presentation using questions submitted through the Q&A feature. Please remember that information provided in this web seminar is not intended as a substitute for your physician's guidance and care. My name is Sarah Rothschild, the Executive Director of the LifeRaft Group. We would like to thank Deciphera for their educational grant for today's webinar. I would also like to thank GIST Support International for their support in promoting this webinar to the GIST community. And now I'd like to introduce our presenter for today. Dr. Neeta Somaiah joined MD Anderson Cancer Center in 2012. She has 13 years of sarcoma and clinical research experience. She is board certified in medical oncology. Her primary clinical and research focus is soft tissue sarcomas, specifically liposarcomas and GIST. She currently serves as the principal investigator of 3 investigator-initiated trials and 9 cooperative group and industry-sponsored clinical trials at MD Anderson Cancer Center, and has in-depth knowledge with extensive experience in the design and execution of clinical trials. She also has multiple ongoing collaborations for translational research, both retrospective and prospective, that help improve the understanding of these rare group of diseases and explore the role of immunotherapy as a therapeutic avenue. Dr. Somaiah has been presented many honors for her expertise and contributions to science, including an Exceptional Women in Medicine Award, and is among the top 1% providers nationally. We know that she's a great clinician, a great presenter, and it's an honor to have her today. So I'm going to now transfer the presentation over to her.
Fantastic. Thank you so much, Sarah. Such a pleasure to be here and also be part of LifeRaft and also GSI, because you guys do amazing work in advancing, just resources for patients and also research in GIST. So that is extremely valuable. Okay. Do you guys see my shared slide view? I can move it to
probably present mode.
Yeah. Is that better?
Yep.
All right. Okay, so let's get started. Sarah already introduced the topic of our discussion today. And we have some disclosures. I do get research funding by running these trials from the listed companies here. I also serve as advisory growth consultant for the companies that are listed here. Additional disclaimers, just because we're talking about a drug and a clinical trial in a space where that indication is not approved, that ripretinib is indicated for the treatment of adult patients with advanced GIST who have received prior treatment with 3 or more kinase inhibitors, including imatinib. So basically, it's approved in 4th line. The recommended dose of ripretinib is 150 milligrams once daily, with or without food, until disease progression or unacceptable toxicity. Ripretinib is not approved or indicated as a second-line agent for patients with advanced GIST previously treated with imatinib by the US FDA or any other global regulatory agency as of yet. Portions of the slide deck represent a scientific presentation by Sebastian Bauer from the ASCO, which is our American Society of Clinical Oncology plenary series that was presented last year in January. Oh, there we go. So this is the study design. We'll come back to it, but this is ongoing. It's a phase 3 randomized, and randomized means you could be selected to go on any one of these arms, global, multi-center, open label study. So patients with advanced GIST, that means you have active disease that you were previously being treated with imatinib. So anybody who's on imatinib that harbors an KIT exon 11 plus 17 or 18 mutation confirmed by central laboratory ctDNA analysis. And we'll go more into detail with it, but just to remember, anybody with GIST who is currently on imatinib and then is having any signs of disease progression on imatinib will be eligible for screening for this study. And we'll go into who will be eligible or not in a bit. The randomization, which is this distribution, is 2:1. So you have a 2 times chance to get on ripretinib, which is the test arm, which we're trying to get into the second line, and compared to sunitinib, which is the current standard in the second line treatment. The study hopes to treat approximately 54 patients, which is a small number because we expect the difference to be wide. And for patients who progress on sunitinib, will be allowed to cross over to ripretinib. So everyone will get a chance to be on ripretinib, on the study. And the treatment will progress until disease progression, unacceptable toxicity, or withdrawal of consent for any reason by the patient. What are the endpoints of the study? So every study will have a primary endpoint around which the statistical design of the study rests. For this particular study, it is progression-free survival. So we'll go into more detail about it, but it just means from the time of starting the study, how long do patients stay progression-free? Means their disease is not progressing on the drug. Secondary endpoints include objective response rate, which as the name suggests, how many patients are having shrinkage. And in clinical trials, we have specific definitions where you basically mark out the tumors that you have. You select up to 5 lesions to be primary, and then you look at what is the percentage they shrink in the longest diameter. And if it's 30% or greater, you qualify as having a response. So, that's objective response rate. Overall survival also is an endpoint, secondary endpoint. Safety, important secondary endpoint. Patient-reported quality of life, trying to see how do patients do with their day-to-day on either drug. Disease control rate, which is basically a combination of the response rate with how many patients are having significant shrinkage, but also, the patients who have stable disease might not have as much shrinkage, but are deriving benefit from the study is also important. And so the combined rate of the stable disease plus responders become the disease control rate. Time to progression, duration of response, which means once you respond, how long do you actually stay in response? And the time to responses, when you do have that significant shrinkage, how much time does it take to get there? So these are select inclusion criteria. It is for adult patients, 18 or older, which is the majority subset with this particular mutation subtype. So you need to have a histological diagnosis of GIST with co-occurring KIT exon 11 and 17, or exon 11 and 18 mutations confirmed by central laboratory ctDNA analysis at pre-screening. So everybody who is progressing on imatinib, either if you don't know your baseline mutation profile, which hopefully is very few patients, because most patients should know what their tumor expressed at baseline when they first started imatinib, or if you are known to have an exon 11. So either you don't know or you know you have an exon 11, you would be eligible for the screening where you get this blood test and determine what is the second mutations or what are the additional mutations you have in the blood. Any patients with advanced GIST with progression on imatinib, again, you need to have received imatinib, but no other TKI or no other oral therapy for GIST prior to enrollment. You must have at least one measurable lesion, and this is when we talk about how we look at lesions that you have or tumors that you have in the body. Measurable is something that's greater than 1 centimeter majority of times, and this should be within 21 days prior to the first, for you starting the drug on trial. Again, performance status should be 2 or less, and for patients, what does this mean? It means that you should be able to self-care, to self-care and be up and about for at least 50% or greater time during the day. You might not be able to do all work-related activities. So that is what 2 specifies. So if you're not able to do self-care completely, then you will not be eligible. Resolution of toxicities from prior therapy to grade 1 or less. And again, every time you have a side effect, we have a way of grading it. So as you would, imagine a low grade or grade 1 means it's minimal impact and usually doesn't require any intervention. So we do want toxicities to resolve before you get onto the study. Select exclusion criteria, or again, if you have, we're unable to confirm the 11 and 17 or 18 mutations on the blood test, then you would be what we call screen, you would not be screen positive, so you would not go on the study at that time. If you do have a history of exon 9 mutations, which is what your baseline mutation profile was before you started imatinib, or you have known presence of exon 13 or 14 mutations, then you would not be eligible for the study. Treatment with, and we'll look at why this is the case. Treatment with any other line of therapy other than imatinib also would exclude you from the study. And if you have a concurrent malignancy, because it becomes difficult to assess what might be getting worse, then you're not eligible. And if you have known active brain metastases or central nervous system metastases, then you're not eligible. As a first step in the study, patients will receive a ctDNA test, which is basically a simple blood test to find out if they have the eligible mutation type. The study doctor will then review the additional participation criteria, which we just talked about. We talked about select criteria, but they'll go through the whole list to determine if you qualify for the study. And if you do qualify, then they will set you up for the cycle 1, day 1, we call the start of the drug. And once you start, the treatment is in 42-day cycle, so it's 6 weeks. And patients will need to come in approximately 3 times during the first cycle. So there will be visits you'll have before you actually get on the study, but once you start, 3 times during cycle 1, which is the first 6-week period, 2 times during the second cycle, which is the next 6-week period, and then thereafter, once every cycle, when you come in to get your drug and make sure all your blood work and all that is appropriate. Travel support may be available for patients on the study. So we do realize every site or every place that you're getting treated at will not have the study. So you will need to go to a different study site, and the travel costs might be reimbursed. Currently, there are 14 sites across the US participating. There are also multiple international sites. The full listing is in that website and can be shared later. Going more into depth of the study rationale. So this is like a graph of or a picture of the lipid bilayer, if you remember from biology. So that's like a cell membrane. And then you have this receptor, and we're focusing on the KIT receptor because that's the mutations we're looking for in this particular study. For KIT is the most commonly mutated gene resulting in this abnormal receptor in patients with GIST. When we talk about primary mutations, that means the mutation that was responsible for the tumor to arise in the first place. So early disease is usually driven by primary mutations, and majority of them are in exon 11, which is the location on the gene. And then what it transcribes ultimately is this portion of the protein KIT receptor in what we call in the blue, as you see there, 70% of patients will have that. We call that the juxtamembrane domain, which is close to the membrane. And then 10% of patients will have an exon 9 mutation, which results in an abnormality in that location, in that extracellular domain right above the cell membrane. And then what you have here in I think it's purple. I need to get my colors straight, but the bright purple magenta color is where you could have an ATP binding pocket in exon 13 or 14, and exon 17 and 18 form the activation loop location of this KIT receptor. Now, those tend to be secondary mutations majority of the time. So when we call secondary mutations, it's also known as resistance mutations to imatinib. So when you're progressing on imatinib, the cause of that progression could be development of these additional changes, which are then, could be in these loci. Sometimes you could have multiple, but sometimes you could have just one. So this is INTRIGUE was the predecessor study that, based on the data that we found out from that, made it, obvious that this INSIGHT study needs to be run to see which patients would benefit the most in second-line treatment. So the initial study was INTRIGUE. It was a phase 3 study again, where which compared ripretinib to sunitinib in patients with advanced GIST who had progressed or were intolerant to first-line treatment with imatinib. So as you can see here, the big difference was there is no mutation requirement on this particular study. So everybody who were progressing on imatinib were allowed. The results of the study showed it was designed to look for superiority of ripretinib, but what it showed is that ripretinib was not superior to sunitinib at increasing the length of time that patients survived before their cancer got worse, like before it progressed. The median, when we call median, so you look at this graph on the side, this is a graph we see in a lot of our study results where the y-axis is the survival probability. In this case, it's like surviving without progression probability or percentage. And then on the x-axis is the time in months from the time you started the study. So if you look at the time point 0 on the x-axis, that's the time everyone's entering the study. So nobody has progressed on the study or on the new treatment because they're just starting. But as soon as they start, you will see the bars, the graph dropping at each time point when a patient has progressed. So progressed or any event which is either death or progression would qualify as a dip in the graph. As you can see, these two graphs, the red line for sunitinib and the blue line for ripretinib, run pretty close to each other, which means there was no difference. And when you look at median, it's basically the middle number of all the patients on the study for what their time was without progression. So for ripretinib, it's 8 months, which means 50% of the patients are below that and 50% would be above that in terms of how much benefit they're deriving. And for sunitinib, it was 8.3 months. And this was not statistically significantly different, as we can see just from the graphs running together. So for INTRIGUE, the inclusion criteria, as we already said, was adult patients with GIST who had progressed on imatinib or had documented intolerance. This particular study had a lot more patients, and they were enrolled from 122 sites across North America, South America, Europe, Australia, and Asia. Because there was no mutation requirement to enter, the patients had a wide variety of mutations. So they were stratified based on, so we did find out what mutations they had, was stratified based on whether they had an exon 11 or an exon 9, or they were wild type for KIT or PDGFR, which means they did not have a mutation in either KIT or PDGFRA gene, or if there were other mutations in KIT and PDGFR. Intolerance to imatinib is also a stratification criteria. Here, the randomization was 1:1, which means there was an equal chance of you getting on ripretinib first or sunitinib. The dosing was 150 once daily, as we have right now for the current study, and sunitinib is also the same, 50 milligrams once daily, 4 weeks on and 2 weeks off. So whenever the patients started on the study, they started on this on-label dosing for sunitinib, but each investigator had the ability after cycle 1 to change the dosing, whether it be continuous or lower dose based on tolerance. The primary endpoint for this was also progression-free survival. And IRR there means by independent, radiology review. So it means it's not only dependent on what the investigator thinks, the scans are also reviewed centrally by a radiologist who's blinded and can tell what's going on, whether it's getting worse or is the same. And in this study, there was no crossover because, this was second line, once you progress, the investigators would decide what is the next best drug for you to go on. Every patient on the study had a baseline, or were at least recommended to have a baseline blood test on which that ctDNA analysis was run. We're doing this as a requirement for the INSIGHT study, but this was done as part of the INTRIGUE study. ctDNA examines the patient's blood to detect DNA fragments from the cancer cells. So we now know that cancer cells will shed DNA, and that can actually be detected in the blood. Very neat. It's also referred to as a liquid biopsy, and so it provides a very minimally invasive but rapid results for mutation status of patients. And you can see the treatment, sometimes some mutations will go down and will be nil, and then you'll see them recur as if the disease is progressing. Again, it's not 100%. You won't detect it in all patients, but the sensitivity and specificity has increased quite a bit over time. Mutational ctDNA analysis following progression in imatinib can provide more insight to resistance mutations in advanced GIST. And the INTRIGUE study did bring that to light quite well when you look at this exploratory analysis. So when we say it's an exploration, it means it wasn't designed as one of the primary or secondary endpoints of the study. It was more of an exploratory endpoint, was that we were going to look at the mutations and see if they made a difference to how you did on ripretinib versus sunitinib. And what you see here is a forest plot. We call that a forest plot in our lingo, where if you're on the right side, the bar on the right side means it favored sunitinib. Patients did better with sunitinib. And if it was on the left side, it favored ripretinib. So if you focus on the yellow rectangle there, which is what we focused on, if you had an exon 11 plus a 17 or a 18 secondary mutation, and they were around 16% patients on the ripretinib arm initially, and 24% on the sunitinib arm. You could see that the median PFS, so mPFS, was 14.2 versus 1.5 months on sunitinib. And conversely, if you had an exon 11 plus a 13-14 secondary mutation, if you had any evidence of a 13 or 14 secondary mutation, then you did better on sunitinib with a 10.9 median PFS versus an 8.1 median PFS on ripretinib. So looking at this a little bit more in detail here on this graph format, the previous graph I showed you how the lines ran together, which means there's no real difference. Here you can start seeing that there is a separation of the graph. So the bigger kind of a car that you can run through, if it's a truck, then the difference is even bigger. And if it's a very tiny, narrow difference, then the difference is not as significant. So as we could see, for now focusing on the population of KIT exon 11 plus a 17-18 population, excluding anyone with either exon 9, 13, or 14 mutation. In those patients, again, there were around 27 on ripretinib and 25 on sunitinib initially. You can see that the median PFS with ripretinib, which is in that blue line, is much higher. So a lot, compared to sunitinib, where patients are progressing fast, so that the red graph is dropping down quite quickly. And when you look at the actual median numbers, the median number for progression-free survival for ripretinib was 14.2 months, and it was 1.5 months for sunitinib. And when you look at the graph on the right for the median overall survival, here, ripretinib was not estimable, means we hadn't reached the median yet. By the time this analysis was run, majority of patients were still alive and doing well, or not progressed. And in the sunitinib arm, the median overall survival was 17.5 months. And both these graphs the difference was statistically significant. However, this is an exploratory analysis. So no concrete conclusions, and you cannot make FDA changes or label indications based on exploratory analysis. This is just this is hypothesis generating, and we need to test it further. So now you can see why the current INSIGHT study was designed, because this difference was significant, and we wanted to make sure can we bring the second line treatment and tailor it according to the mutations you have, because we know that can make a difference to get on the right drug early on. Looking further at the exploratory analysis, looking at response now. And again, remember I said we measure lesions and we look at percentage drop in the size of the lesion. And if it's more than a 30% drop, then it qualifies as response in the clinical trial lingo by RECIST. On the left-hand side, you see the sunitinib, what we call, this is the waterfall plot. And on the left-hand side is ripretinib. So at the 0 mark, which is the middle line, that's the amount of tumor you have when you enter the study. So we made measurements, and that's your baseline. Then as you go through the study, at best, how much did your tumors drop at the best time point of being on the study is determined by how deep that drop is. So, and if your tumors got bigger on the study, then it's how, the bar is, how high the bar is above that 0 time point. So being below is good because it means the tumors are dropping, being above is not good. So if you look at just that population of exon 11 plus 17 and 18, you can see for ripretinib, 44.4% of patients who met that response criteria of greater than 30%, and majority of patients had a dip in their tumor measurements, but only a few patients had the bars going up. Whereas when you look at sunitinib, it's quite the opposite for this particular set of patients. Again, side effects are important because, this is something we want you guys to be on for a long period of time, take every day, and compliance, all of that is measured with how you tolerate the drugs. So when you're looking at any grade 3 or 4 drug-related TEAEs are called treatment emergent adverse events. So adverse events is any side effect reported after you started the study. And when sometimes you could have a side effect completely unrelated, you ate some bad food and you had a, GI distress versus something that is related. So when we call treatment emergent, we are looking at basically all things reported in the study, and we are not really saying whether it is truly related to the drug or not, okay? When you look at ripretinib, now when you talk about grade 3, 4, usually that means a higher grade of toxicity, which required intervention, either a change in the drug, hold of drug, a dose drop, or needed intervention to treat that particular side effect. You could see for ripretinib, the grade 3, 4 treatment related also, sorry, these are treatment related. So we know that was associated. The investigator said that this side effect is related to this particular treatment. That was 33% for ripretinib and 50% for sunitinib. And when you look at, again, drug-related treatment emergence, SAEs, which is serious adverse events, which usually is either you got hospitalized or it meets certain criteria for it being, or hospitalization or prolonging of a certain hospitalization. That was 3.7% for ripretinib and 13% for sunitinib. Now, looking at what, that's again small, very small percentage on either arm. When you look at all grades, which means, grade 1 minor versus a serious side effect, when you look, at the differences, I'll point out ripretinib first. Alopecia, hair loss, is very common with ripretinib. You end up with a a little fuzz left behind it. Over time, depending on how long you're on it, your hair might go back, but that is a definite change from imatinib. For sunitinib, you don't have as much alopecia, like full hair loss. You might have a little hair thinning, majority of cases, but your hair color changes. When you look at ripretinib, again, the myalgia risk, which is muscle aches and muscle spasms, was a little higher, which is like 44% or 30%. And again, sometimes they were mild, didn't require intervention, and sometimes might need intervention, compared to 13% or 8.3% for sunitinib. In terms of hypertension and PPES, which is palmar-plantar erythrodysesthesia, which is what we call hand-foot syndrome, where you get painful calluses in your palms and soles, and that can be debilitating in terms of functioning, especially depending on the grade. That was what we always say is higher with sunitinib. So the plantar or the hand-foot syndrome was 42% for sunitinib, and the hypertension was 50% with sunitinib, compared to the 33% for ripretinib and 37% of the hand-foot syndrome with ripretinib. Fatigue is also kind of equivalent for, it was reported 48% of the time with ripretinib, 38% of the time with sunitinib. Constipation, again, we worry more about diarrhea in this situation. And, when you look at diarrhea, 22% with ripretinib and 38% with sunitinib. Again, most people will do just fine, but some will need intervention with Imodium or other drugs. So getting this early intervention is important. The rest of the side effects not. So really the ones are the hypertension and the hand-foot syndrome, higher with sunitinib, the hair loss, and the myalgias, muscle spasms, a little higher with ripretinib. So this is the INSIGHT study, again, the one that we're focused on today. We looked at this, the schema before. Again, to reiterate, one prior line of imatinib. So if you're on imatinib and now there is concern for any tumor growth, you could be screened for this study as long as you have an exon 11 baseline mutation, or you don't know your baseline mutation, then you can be screened by a ctDNA, which is that blood test, so that you'll sign a screening consent, and you will get this blood test drawn. And if it's positive, then you get randomized, which is 2:1, higher chance of getting on ripretinib when you have the exon 11 plus 17 or 18 mutation subtype. And then you'll go on, and when you have progression, if you're on the sunitinib arm, you have the option of crossing over to ripretinib. For additional information regarding the INSIGHT trial, you could, well, we didn't put my information in here, but you can contact on the clinicaltrials.gov website. There is, there is listing of all the sites where the study is open, and there'll be contact information for people in that particular site. I'm sure LifeRaft can let you know how to get in touch with me if you need to. And that is the LifeRaft group address over there for you to look at. And that is about it. We slated a lot of time because sometimes there are a lot of questions, and through questions, the understanding of this gets a little bit more deeper, even if it's a repeat of what I have said. So if Sarah, if that is okay, should we move on to the Q&A session?
Yes.
Good.
Thank you so much.
Yeah.
Thank you for this wonderful
I
presentation.
could, yeah. Would you want me to keep the slides up to refer back based with questions, or should I just stop sharing and can go back to it if the question requires?
Let's stop sharing and then let's go back to it if needed.
Okay.
Okay, great. Thank you. So the first question is, how long does it take from the screening time to starting treatment once eligible?
Once eligible. So if you're saying once eligible, then you have your blood test back. So, well, I'll go through the whole timeline because that will be easier. So if you are identified as somebody progressing on imatinib, and you're at a center where the study is open, they can have you sign a pre-screen consent where they can send your blood to get that mutational analysis done. It is coming back, and my patients, I can tell you, it's usually within a week that you get that blood test back. They say you could wait up to 2 weeks if there's a problem or something, but usually less than a week. Then once we know you're eligible, then starts the screening phase, and that usually has to be within 21 days to starting therapy. So then you get they go through all the other eligibility, make sure you're eligible. They will have you sign the consent for the actual treatment, and then bring you in with a fresh set of labs before you do your what we call cycle 1, day 1, the first step. So this can take, I can tell you from the time of identifying a patient and sending the blood test, it usually takes around 2 to 4 weeks to get them on starting cycle 1, day 1. But it can be, it's fairly quick. It's not more than that. It's usually a couple of weeks.
Okay. And how often will participants need to visit the clinic or hospital for assessments and screening during the trial period?
So one thing, good about the study. So again, there are visits before you start, and there are visits once you start the study. So the visits before, the great thing post-COVID has been a lot more remote consenting. So at least at my center, we do have the ability to. So if I've identified you or we've seen scans that's getting worse, we can contact you. We can have you sign the pre-screen through a virtual visit. And there are a lot of other centers who also have this capability. I think the study is also working with an external agency where they could do this pre-screen for you so that they can identify at your local site and then send you off to the sites where the study is open if you qualify and you're positive. So there is that ability of pre-screening elsewhere. And so those visits can be done virtually, and then you have to come in at least once with all the blood tests to be done, and then see the physician before you actually start cycle 1, day 1, to make sure you're meeting all the eligibility criteria. So once you start that cycle 1, day 1, as we said, like the first cycle, I think it is up to 3 times you have to come in on cycle 1, and then it's 2 times the next cycle, which is the 6-week cycle. When I'm saying cycle, it's 6 weeks, because you usually get the drug dispensed for that period of time. And after cycle 3, you get the drug dispensed for 6 weeks, and then you come only once every 6 weeks. And the drug is provided by the study, so you will pick up the drug on that visit date when you go in.
Got it. That was very helpful. I know you touched upon travel support, but just want to go back to that regarding, in general, financial assistance for the trial. There was a question about lodging and other costs that might be supported. Could you speak to that?
Yeah, I mean, again, there might be a little difference because these are all negotiated based on the contract with the sponsor. In this case, it is Deciphera that's sponsoring the study. So, but there is travel reimbursement. So we tell patients to keep the receipts for their flights, their lodging, their food receipts, and then they get submitted through the research coordinator to this company that handles the reimbursement. So there is travel support, but remember that the travel, this comes up sometimes because patients might come wanting to be on the study. The travel reimbursement usually only is eligible after you've signed consent for the study. So then all the receipts after that would be covered.
That's a very important point. I'm actually going to just circle back to my previous question. As you were referencing cycles, somebody wanted to know how many cycles altogether?
So, with gestations, a lot of them have probably heard this. If you have disease that has not been completely resected and your risk is low, most patients who have advanced disease, which means it is spread or it's not resectable, have to stay on TKI suppression, for as long as possible, right? So as long as you're tolerating it, don't stop it, because when you stop it, it progresses. So the same applies to these drugs. You will be on the study, you will continue the cycles for as long as it's working. But in a clinical trial setting, so as long as it's working for you, you will continue. And then it happens, well, how long do I keep continuing on this study and having to keep coming back, right? If it's working. So the goal is, if the study completes the enrollment of the 54 patients, and then you have to get the readout of the study where we know if it was a positive study, which, we're all expecting, but we have to look at the data before we decide anything. So when we see the readout, then the data can go to the FDA, and there is an approval process for the second line treatment. So then it's potentially conceivable that, usually most companies will continue to provide the drug for you when you're on, if you're on the study and benefiting, but at some point when it gets approved, there might be a transition to commercial supply. So those things are not, 100%, I can't speak to exactly what will happen, but most of the time you will, if it's benefiting you, you will continue on the drug, and if not, you will be taken off the study and put in something else, yeah.
Okay, more questions are coming in. The good thing is we have time to answer them.
Yeah.
Okay, so the next question is, would there be crossover on this trial?
Yes, which is very important, because, as you can see from what we learned from the INTRIGUE study, so patients who have this mutation subtype might not do, or at least most of them won't do very well on sunitinib. So if we do expect patients to progress on sunitinib, they should be an option. So that is the attractive part about the study, is that though, yeah, if you get on sunitinib, what do you do? Well, if it happens to benefit you, then great, you stay on the study. But if not, you will cross over and be allowed to switch to ripretinib, which will also be provided. You'll still continue on the study, and it will be provided by the study.
Okay. There's a question that came up about exon 11 is known. Well, why should I discuss checking my eligibility of the INSIGHT trial versus just going into the second line student? What is the benefit of doing a trial rather than just taking a medication that's already approved for GEST?
Yeah. So, and the thing is, we're always trying to make it better, right? We know we have a second line approval. Well, we got a fourth line. We need more drugs, because unfortunately, once you get to second line, there is disease. There's always, risk for resistance developing and growing. So we're looking at what are the ways, firstly, getting new drugs and having additional options, but also, is there a way we can delay that progression? Can we delay progression? Can we delay, if you're controlling the disease better, will that mean less secondary mutations in the future? I don't know that, but if you get longer on your second line, will that make it better, because you won't have as many mutations later down the line? So the earlier you get on a drug that is working, we all know that's better. So if your disease is progressing, so earlier you get on a drug that's working for you, the better it is, because then you'll have control. So the reason to participate, the reason to do the study is we're hoping that in the future we can, try to intervene with a drug that is more likely to work, where the chances of response, like we're saying, if it's just second line and it's all comers and we don't know what mutation profile you have, then from the studies, we know the chance of a response with sunitinib is usually lower, like, it's less than 10%. Doesn't mean you don't get benefit, you could have stability, but the extent of response is lower than what you get with imatinib. So it would be great to be able to say, well, you have this mutation profile. If I give you Sutent, now your chance of response is great. It's like close to 50%. Or I can give you ripretinib if your mutation profile is this, and your chance of response is greater than 50%. And in second line, if you don't have too much disease, it is conceivable that if the disease is limited and if you have a great response, you could go on to get more ablation, something where the duration of that response can be longer. So, the goal is to prolong life, improve quality of life while prolonging. And a lot of times the quality is also related to amount of disease you have. So if you can decrease that disease burden, the side effects are better. So the reason to enroll would be there is a chance of being part of a study where your mutations will guide your treatment and you have a chance of response. But a lot of that is then also personal, right? What is the burden of being on the study? That is, it's an individual thing. As we say, we can try to make things easier. You can provide travel reimbursement, but everybody has different challenges in their life. So if you cannot get on the study and you're starting second-line Sutent somewhere, that's fine. I just, making sure that you're aware and know that the knowledge of that mutations that you have could impact your outcomes to these drugs is important. So yeah, I think I spoke long because it's a little passionate. I'm like, this is how we can advance things a little bit. Yeah.
It was a very important question. So it was important, I think, to have that lengthy explanation. But I want to go deeper into the topic of mutational testing because some questions came up about that. So would I still qualify if I've already undergone mutational testing?
So there are two types of mutational testing. One is whether your tumor tissue was tested, that's the tumor mutations, and then it's the liquid biopsy, which is that blood test. So of course, if you've had mutational testing, which as I said, hopefully most people do get it up front when you have advanced disease, you still qualify as long as you have a known exon 11 mutation, or sometimes if they didn't find a mutation, it's possible that the test was erroneous, so I wouldn't exclude those patients. The only time I would say if you've had mutational testing and you wouldn't qualify for the INSIGHT study is if you are known to have an exon 9 mutation, right? Or if you're known to have an exon 13 or 14 mutation already on your mutation testing, then it will be of less value because the chance that that will disappear is unlikely, right? Or you would suddenly get an exon 11. So those patients I wouldn't screen, but patients who don't know and are coming to me or are known to have an exon 11 only, we can screen them at time of progression. So your prior testing does not disqualify you from being screened.
Okay, so that sort of feeds into another question that came in about how will I know if I have a secondary mutation? If imatinib stops working, did that automatically mean that I have a secondary mutation?
It might not. So again, secondary mutation detection rates do vary. So of course, if there is a tumor that is progressing, if you biopsy that, you will get what's happening in just that, tumor. And, there is a likelihood you'll detect additional changes other than your primary exon 11 or exon 9 that's likely, causing the progression. But a lot of time, not a lot of times, I should say, majority of the times we will detect something secondary, whether it be in KIT itself, or sometimes there could be additional mutations in different pathways that might be causing the resistance. So, there you have to put our brains together, say which ones, which of the drugs would be more useful, and you wouldn't qualify necessarily for this study. But, on a liquid biopsy, the detection rate is, the percentage of detection of these mutations might be a little bit lower, but the reason it is much better is because it allows you to get a much broader view of the mutations profiles of all the tumors that are in your body, or any cells that might be anywhere. If they're shedding, then the liquid biopsy has a greater breadth of detection rate, even though sometimes you might not, get it. So yes, if you're progressing on imatinib, you should get tested, but it's not 100% that you will always detect one of these secondary mutations. You might detect other pathways. It's also possible, I have had patients who are not very compliant sometimes with their imatinib, and their progression is due to the compliance or because of being at a lower dose because they couldn't tolerate the drug, or because you have an exon 9 mutation, primary mutation, and we know that imatinib at the lower dose of 400, which is usually started, doesn't work as well. So those patients, you might not have a secondary mutation. It's just that you don't have enough dosing of the imatinib in to control the disease. So your physicians might actually change the dose.
Okay. And you did refer to liquid biopsies. So the question was, will mutational status for admission to the trial be determined by liquid biopsy in all cases, some cases?
All cases. So for this, there was a bit of debate. We were like, well, if someone did end up having a biopsy, shouldn't we, include those? But just for consistency, and since we're trying to get FDA approval, you don't want any, flags, because what if you biopsied one lesion and the second lesion has a different mutation profile and you don't catch it? So they wanted only liquid biopsies. So you do 100% of patients for their pre-screening will require a liquid biopsy for ctDNA for eligibility.
And then, somebody had commented about, well, studies show exon 17 response to Stivarga or regorafenib more than sunitinib. So how would this affect a patient if they end up on the sunitinib arm of the INSIGHT trial?
So if they end up on the INSIGHT, if they end up on sunitinib, of course, again, the thing is, we still want to see either clinical or radiographic progression. So if there's any concern that patient's clinically progressing, you could do a scan sooner, or if basically at the first time point at 6 weeks, you do a scan. And if there's any growth, then you go on to cross over to ripretinib. I mean, if you choose not to get ripretinib and go to regorafenib, that's fine. But so this study is not testing for whether regorafenib is superior or ripretinib is superior for exon 17. It is testing whether ripretinib in the second line is superior. There are other studies that might be looking at regorafenib in this setting in the second line. But looking at the side effect profile and the prior study with INTRIGUE, this design made the most sense. You could still access, if you go on the study doesn't involve regorafenib, but you can always get regorafenib after. There are, some patients who might respond on that if they don't respond on the study. Is there differences in tolerance and how each patient metabolizes the drug?
Okay, thank you. A question came in, can this drug be taken to prevent GIST recurrence?
So currently, no, and I'm not aware of any preventive studies with ripretinib. So the only drug, when you're talking about prevent, is after you've had a diagnosis of GIST, it has been surgically resected, and there is no evidence of disease on the imaging. What do you do to prevent this GIST from coming up again? Now, there is data, there are nomograms, there is, risk stratification criteria. LifeRap group has that on the website as well. So you could look at what is your risk of recurrence, right? Because there's always a risk benefit. You don't want to, everybody getting surgery, 100% you put them on a drug, the cost of that drug, the side effects of the drug might not be worth it for patients who have a very low chance of recurrence. So in the community, we think if it's 20% or higher risk of recurrence, it's worth a discussion. If it's 50% or higher, then majority, we would absolutely encourage you to get on imatinib for preventing the recurrence. Again, imatinib works in majority, because as you said, majority have 70% will have the exon 11. Then there's some other mutation subtypes which also respond to imatinib very well. And for those patients after resection, imatinib has a relatively great side effect profile for majority patients. I know some patients do not tolerate it at all, but most patients will. And the side effect profile and the amount of benefit you get has what led to studies, and there were studies done looking at adjuvant, what we call adjuvant after surgery, to prevent recurrence. So imatinib is the only drug that is approved in that setting right now. I would not use ripretinib in that setting as a general rule, because that is not approved in that setting. And there will have to be a benefit of doing. So as you can see, we have primary mutations, which runs drives your early disease, and then you have secondary mutations. So the likelihood of having a primary mutation that is responsive to imatinib, is much higher, and you're less likely to have secondary mutations requiring any of these second or third or fourth line drugs in that setting. I can tell you that patients on imatinib, if they do progress or if they don't tolerate it, then it goes into a more in-depth discussion about, is there, because even when you're saying prophylaxis, there's different ways. You could have just a tumor that was removed versus you had a tumor with 3 or 4 other nodules that were also removed, and now you're without evidence of disease. So the risk for both those is very different, right? Somebody who had tumor rupture, so we know there is tumor there, versus we just remove the tumor and we don't expect. So then there is a different discussion for, if you didn't tolerate imatinib, for patients who have just almost 100% chance of recurrence, would you treat with something else as a prophylaxis? And that is an individual discussion to be had.
Speaking of tolerating, if a patient is on the sunitinib arm and can't tolerate the 50 milligram dose, would their dose be decreased or would they be crossed over?
The dose would be decreased. So, though sunitinib gets a bad rep, there is, it's about dosing. You could get to 12.5 milligrams and you end up tolerating sunitinib. So the dose reduction would be the first recommendation. And then I can talk about my thing. It may not be everybody else's, but if it's 50, 4 weeks on, 2 weeks off, you're not tolerating it because by the time you get to the third week or even fourth week, it's getting too much with the hand-foot. I usually drop the dose to 37.5. Depending on the rate of the toxicity, I might even drop it lower if it's really bad, and also looking at the blood counts. And then I tend to, if you're tolerating it at the lower dose, then you could even take it continuously. You don't need to take the 2 weeks break off. Now, if you're just not able to tolerate it, you know that you will be at the lowest dose, you'll get a scan, you'll progress, and then you would cross over. But I think the way the study is written is you do need to have that scan showing growth before they will cross you over, just so that it doesn't, otherwise people would say, oh, it's possible that you were responding on sunitinib before you went on to ripretinib. Yeah.
Okay. And then let's talk about the dosing for ripretinib. Is there a significant difference in survival between the 150 milligram per day versus the 100 milligram per day?
So a good question. To be honest, I don't think we have an answer to that. I don't think we know. Well, when we do the phase 1 study with dosing, so we had patients on lower doses of ripretinib in the phase 1. We also had patients with a higher dose of ripretinib. So we have gone up to 150 BID, even in the other studies with ripretinib. Ripretinib dose reductions were actually quite rare, lower compared to what you would see with dose reductions on sunitinib. But the dose reduction, if you have a significant myalgias and you need to reduce the dose, 100 milligrams was used. So it was allowed on the study. Patients who had dose reduction also derived benefit, but we didn't have a way. I don't think we've compared the 150 dose and those who reduced the dose. What was their difference in survival? In terms of the PK, the way they came with the 150 milligram once daily dosing compared to the 200 and the 3 and 4 was that the PK, the levels and the level of suppression of the various mutations that were seen seemed the most optimal. So the side effects were manageable, and the levels that the drug reached in your body were sufficient for the IC50 to cover, or the level that's required to cover the mutations that are most commonly seen.
Okay. How soon is the timeline for ripretinib to be the standard second line of defense for GIST patients with exon 11?
So it'll be for exon 11 plus 17 or 18 based on the study if it is positive. So the timeline will depend on how fast we enroll on the study. So one of the things of, we don't talk about, because, when we're taking patients, we're just interested in your outcome. But in general, when patients participate in studies, you're also working towards the global betterment of data and learning about your disease, of course. And if it's a study that also can potentially benefit you, it's a win-win, right? You're got to be, once they complete enrollment, we also have to see the outcomes, which is, how many patients, once you enroll them, when they progress, that's when the events get counted, and it has to meet, be able to come up with the statistical analysis. It usually takes, the minimum would be a year for a readout if you enroll everybody in the next month, which is not happening. But, it's like, so enrollment, because we have to screen patients. So the more screening we do, I would say a couple of years, could be 2 years before it gets approved. But this is all speculative. Again, depends on how fast the study accrues and how fast we get the readout. The interesting thing is, because the level of difference we saw on INTRIGUE was quite high in this subgroup. When the difference is high, the events will happen faster, and it is likely we might get an early readout where it says, well, patients are doing much better on this. We need to, get this early readout and then go to the FDA. So it's possible in a faster timeline as well.
And then how does that relate to these NCCN guidelines? Not everybody might be familiar with how important those are. How would that sort of translate there? And I just want to point out, somebody who must be from China who's on the line was saying that apparently the Chinese Society of Clinical Oncology already recommends ripretinib as second line for exon 11. So things may differ between country in terms of, I guess, what's already out there, but at least for the US, you could share your thoughts on that.
Absolutely. So, there are just different levels of approval, and those levels of approval also guide insurance reimbursement for these drugs. And when the FDA is looking at data, which is when you say it's an FDA-approved drug, and then most insurance companies will have to cover it, it's based on the level of evidence. So usually it's a phase 3 study, which is randomized. So you're looking at the difference between the new drug versus what is the standard in that setting. Or it could be a rare subtype where the outcomes are so good and there is no standard, then they might do it without a randomized study, but that's the FDA approval. Now, NCCN is an agency that lists guidelines that almost not only the US, but the world basically follows in terms of how to approach treatment for different types of cancers. And it is formed of a committee with different, NCCN centers with expertise in disease. Will have a representative in this group that then reviews data as it comes. Sometimes they review data on a more expedited way than the FDA would. So if there is data, especially in rare tumor types, where we know that we won't always be able to get approval fast enough, but if there is enough data supporting the use, they might put it into the NCCN guidelines. And this is a big help because that also is another means of getting insurance companies to pay for this drug. Because unfortunately, none of these drugs are cheap. When you're on the study, of course, it's provided, but after that, the sticker shock of the value of non-insured like medication can be quite high. And it also, it's not only, getting reimbursed, but it is also the group of people are looking at the depth or the, not only one study, but looking at the wealth of data out there saying, what is the best thing for patients? We've struggled now in the NCCN guidelines. We have that you have to, when you're starting any TKI, you should have mutational analysis done, right? It's in the NCCN. You should have KIT, PDGFR, you should have SDH, IHC, because these guide treatment options for patients, and it is important for patients to get this done. Similarly for drugs, right now in the NCCN, it actually lists sunitinib as second-line therapy, but they, based on the INTRIGUE, the INTRIGUE study, they do have patients who cannot tolerate sunitinib. They could get ripretinib as second line. So that is there in the NCCN guidelines. It's still not easy to get. So the thing is, if you don't do the study and just rely on that, it won't be ultimately covered, everywhere. It won't set the guidelines. So this study would actually get it more formally included in NCCN guidelines for a specific mutation subset. As I said, regorafenib could work, so you could go to that next, but all the sequencing of these drugs would be interesting. Now, I didn't know that in China it's listed for exon 11, you could get it second line, but that's interesting. And the question is then, what happens? Like, if you don't do the study and study it, we don't really know what happens. You have to really collect data. So when you get a second line, what happens to them third line? What happens to this will give, following these patients on the study gives us a lot of information that guides not only the treatment of the patients on the study, but also treatment of future patients. So,
yeah, great.
Sorry, again, a long answer yeah.
Great information, though, very important. Circling back to a little bit more regarding mutational testing, that's specifically on liquid biopsy, a few questions came in. Since liquid biopsy has more breadth, then why is it not used for regular periodic monitoring for progression of disease or response to treatment? Why is it not a standard of practice?
Because, when it becomes, so for it to be standard and accepted, there are two things, right? Of course, it could be, with this, if the study is positive, you probably will get an approval of liquid biopsy as a standard companion diagnostic where you need that liquid biopsy for patients progressing on imatinib in order to then, because it is going to make a treatment decision change. So far, in any of the treatment criteria for GIST, we have imatinib, sunitinib, regorafenib, ripretinib, and then we have avapritinib if you have a PDGFR mutation, but that's on your baseline, and that baseline is usually on tumor biopsy. When you're progressing, the sequential blood draws for liquid biopsy is done. I mean, it's done at a lot of academic places, including mine, at certain time points when we feel that it might be important and we're actually gathering data, and sometimes it could affect your care. But so far, it hasn't been important in selecting what your next line of treatment is going to be. If you already know that you don't have PDGFR, and so you don't exon 18, so you don't need ripretinib. For all the others, we go through the whole sequence of drugs. So it hasn't been important, but as more data comes along, as we keep doing these, we might identify resistance mutations that will help guide us with the next trial that we need to do for patients and what drugs to bring in and how to cycle it. So I would say that, there is a cost. We talk about the pinprick of a blood test might not be too much for a patient, but for some patients it can be a lot. So, and the cost of doing the test is also high. So if you're doing it because you feel it will give you data that can be helpful, then it gets done. But otherwise, doing studies like this and actually proving that we can alter treatment and outcomes for patients based on that liquid biopsy, that's what will get it covered. And
Yeah. And somebody had asked about, well, what if it doesn't pick up the secondary mutation? Are there other ways to pick it up? And that would be like the biopsy from an actual tissue sample.
So it would. If there was one area progressing, it might not give you the whole breadth of mutations. But also know that liquid biopsies, again, not 100% sensitive. So they say that they could get up to 90% of sensitivity, but sometimes it's lower than that. So it's like in that 70 to 90% range. So if it's negative, actually per the study, that is a point on the study that there is one, you could get screened a second time if the suspicion is high, like you had exon 11, but the blood test didn't pick anything up. Actually, you have a case like that where I'm going to discuss whether I should rescreen at a different time point. It's possible that the amount of disease was not enough, the shedding was not enough, and you didn't detect it. So for the study, you could get screened again with another blood test. But otherwise, again, it depends on your physician. If it is going to make a treatment decision change because there's another trial or something like that, then they might get it done again. Or if they really want to know, they would get the tissue to test for it. Right now, getting a repeat biopsy when you're progressing, again, is not standard of care. Because a biopsy is more invasive. So, it is a discussion between your treating physician and yourself to see if there's a value of doing another biopsy at that point.
But what if somebody's just currently not receiving treatment at an academic institution? Could they still participate in this trial? Their current provider?
Yeah, actually, that's a very good question, because I would say majority of patients are not being treated at an academic center, are being treated locally. So, we get calls from patients. We also get calls from their physicians sometimes saying that. So the reason to spread the knowledge is really to say, hey, we know that there is Gleevec, imatinib, sunitinib, regorafenib, and ripretinib. But if you, and most people in the community might just go down that path. But if there is a study, you could stop, because instead of referring your patient out at the time when you're progressing on all lines, it's better to refer early if there's a study. So if you're at a local place where the study is not open, you could bring it up. I know the sponsors at Deciphera have contracted with other people who might allow for local pre-screening in certain areas. So I don't have full in-depth, so you could get the pre-screening done locally with the blood test. Or you could get referred to an academic center and they could pre-screen you. And then once the blood test is back, you will have to travel to, once you're screened and eligible to start, you definitely will need to travel to the one of the centers where the study is open in order to participate as of now. Maybe the future of clinical trials will be different. It will be decentralized. Or centralized, rather to say, but right now that's not the case. Yeah.
Okay. What would be recommended if blood pressure gets raised while on this medication? Is that a criteria to leave the study?
Absolutely not. I would say the most important thing is, so a blood pressure increase is actually an on-target side effect for both the drugs. More so for sunitinib, you'll see a lot more higher blood pressures than you would see with ripretinib. But it's important, and I tell patients starting any TKI after imatinib, we need to do blood pressure measurements at least once a week. If not, if you have a cuff at home, just keep a diary every day, because the early institution of the antihypertensive, which is the medicine to drop your blood pressure, will allow you to stay on the study and be able to receive a drug that might actually be working. Because if it is having an effect, it's possible it's also having an effect on your tumor. So that by itself, I would say no. If you end up with a hypertensive crisis, or one or two drugs or three drugs are not working, like some people have baseline hypertension, so they're already on 3 drugs, so they might have to end up going on a 4th drug, but or increase the dose of what meds they're already taking. But are there some cases that we might take off because of blood pressure? Possibly. But by majority of cases with early institution of antihypertensives, we're able to keep them on the study or on the drugs if they're not on the study. Yeah.
And if a patient is on avapritinib, is this trial an option? I don't know if maybe the patient.
So if you're on avapritinib, no, this trial is not an option. Now, if you're on avapritinib, you either have a PDGFR exon 18 mutation, or you're getting it as part of, because you progressed on other drugs, or you were on the avapritinib study and it's working for you and you're continuing. Because avapritinib also has activity against exon 17 mutations. So, but you would not be eligible for this particular study because it is specifically for second line after imatinib.
Yeah, and the same thing. Somebody's like, well, what if I'm taking ripretinib now? Also not eligible.
Yeah, you're not eligible for the study. Unfortunately, you have to keep getting a ripretinib sourced by the means you are getting it sourced right now.
Okay. And then I guess, you might have mentioned this, but I think if somebody needed just another articulation about this, just ripretinib is available for those who've sequenced between imatinib, sunitinib, Stivarga, so it's 4th line approved. Do they still qualify for this study if they've gone through a sequence of those
drugs? Yeah, no. If you've gone through the sequence, then you're eligible for ripretinib as a standard of care option after regorafenib, or as long, it's actually 4th line or beyond. So any line of therapy, if you've not seen ripretinib before, that would be standard of care. So it wouldn't be part of the study. So you would not be eligible for this particular study.
Okay, I think we sort of covered the range of topics that have come in. No new ones have come about in a while, so I hopefully we covered the range. There was a question if this is recorded, and yes, it is, and we will be making it available for those who couldn't make it today or just want to re-listen to it. So stay tuned. We will share that recording shortly after our live presentation. But I just want to take this moment and say thank you to Dr. Somaiah. We are so appreciative of your time, the way you explain things. You truly are an incredible clinician, friend, and I think that we're very blessed to have you in the field because it helps, I think, the patient community to know that there's knowledgeable people out there who do truly care about the patient and their well-being and their longevity. So thank you for the time today. And with that, I am going to conclude the presentation and thank you those who attended. Be well, everyone.
Thank you so much.